welcome to our new webinar, Repeat Expansions Diseases Assessment with Whole Genome Sequencing. We are very happy to see so many people online, and we are looking forward to our webinar together. So as always, our webinars are very interactive, and we are looking forward for our communication and for our dialogue during the webinar. So please communicate with us, send us your questions, comments. I will monitor them all, and then we have, after the webinar, 15 minutes where we'll address them all, and everything what we don't manage, we will do online with you. So technical instruction, you just go to the Zoom task panel, and then you press Q&A button. Then you will see the menu, and you are able to enter your text here and then send, and immediately we will receive your question. So, and today I'm very happy to introduce Dr. Najim. He is a Senior Director of Bioinformatics Research in Census. Najim has a PhD in human genetics, and he has more than 15 years of experience in molecular diagnosis of hereditary diseases and cancer. So he pioneered his application of next-generation sequencing in a clinical genetics department of the EU Medical Center in Amsterdam. And whenever Najim gets a patient sequencing data and the patient has a genetic disease, he and his team will always identify it. So we're looking very forward to our webinar with Najim. And I would like to remind you that for sure we provide our attendance certificate, and it's important that you properly fill all your details in the form. So provide your full name, and then you will receive the certificate after the webinar within several working days. And with that, I would like to introduce Najim. And thank you very much, Najim. So we are looking forward for your webinar.
And welcome everybody to this presentation, which is going to be about repeat expansion analysis using whole genome sequencing data. So let's get started. Okay, so let me try to get a pointer. So during this presentation, what I hope to share with you and convey as a message is diseases related to expansion repeats, to repeat expansions. I will show you how this occurs at the cellular level, just in brief, as a brief introduction. And then I will tell a little bit more about the methods that I used to detect those kinds of expansions. And at the end, I will leave the floor for questions, which I will attempt to address. So let me move this a bit up. So repeat expansions, these are genetic mutations that can affect 1 to 6 nucleotide motifs, which are also called short tandem repeats. They are also called by others as microsatellites. These are motifs that can either be present in the coding region of the gene, or in the intronic region, or in the untranslated regions of the gene, which are either 5' or 3'. Normally, these motifs ranges are different per motif, per locus, and that they can range from a single motif up to 80 repeats, which is in the normal range. And this is then depending on the genomic location. And some of the repeat expansions, they can exceed over 1,000 repeats, which is, for example, in the repeat expansion that is related to familial monoclinic epilepsy. So, as I mentioned earlier, there are different repeats that are present either in the UTR region or the exonic region, and this is just an overview of the different diseases or loci that are associated with diseases and the type of trinucleotide most of the time, and sometimes different kinds of motifs, and how they are located at the different locations of the gene. And usually the majority of the exonic trinucleotides, they are either result in glutamine repeats or poly-A repeats. And I will tell a little bit more about these kinds of repeats in a minute. So how do this type of repeat expansion occur in the cell? So briefly, this is a schematic overview, which I will explain in the next slide in a little bit more detail with some animation. So when you have a DNA sequence that contains a motif that is repetitive, during the first replication cycle, what happens, as you may or may not know, that during the replication, there's one strand that is copied continuously, whereas the other strand is copied in segments. So the segment that is copied continuously, you have this helicase, which tries to unwind the DNA. And once it arrives at this repeat sequence or repeat motif, it slips. And then once it slips, a hairpin is formed. During this hairpin, this will result in this particular hairpin forming in the first round of replication. And when the next round of replication takes place, this hairpin will be stretched out and it will be sequenced and then inherited to the next cell. Okay, so I would like to show this video which illustrates this in an animation, which in my case, it was helpful to understand how this works. So let's hope this would work out. I will try to start this illustration. So what you see here, the yellow part is the sequence that is a motif that contains nucleotide sequences. And then we have the DNA, which is in red here. And then this blue part that you see over here, that is the helicase that unwinds the DNA. So we see the yellow part, which is a repeat, migrates into the region where the replication takes place. And once it arrives at the location that does the nucleotide replication, you see that the hairpin is formed. And this will be inserted into the copied DNA. At this time, there is no repeat expansion that took place yet. So during the next round, which is this one, once the hairpin arrives again at this replication location, then this hairpin will be unwound and then it will be replicated and inserted into the next round. So hopefully this made it a little bit clear for you to understand how this repeat expansion has taken place. And now you see that the yellow part has got an additional orange part inserted, which is now the repeat expanded region. So there are different types of diseases, which are over 50, that can be caused by repeat expansions. And the mode of inheritance of this type of diseases can be either autosomal recessive, autosomal dominant, and X-linked, depending on whether this locus that has the repeat is on the X chromosome. And the diseases, in the majority of the cases, they appear at earlier age in a subsequent generation. And this is a phenomenon that is called anticipation. Mainly, there are two types of diseases associated with repeat expansions, some of which affect the coding region, as I mentioned earlier, and most of the time, when a coding region is affected, and this is mainly a polyglutamine or poly-A repeat that is expanded, it is usually a gain-of-function effect. So the product of such an expansion would result in a toxic protein. Later, a little bit more on this. And then you have other expansions that affect either the intronic or the UTR region. And these types of regions, when they are affected by repeat expansions, they mainly affect the expression repressor. So you have less or no protein made from this particular for this particular gene. Just to give you an example, and this is just a set of diseases that involve repeat expansions, and I tried to order them based on the number of motifs or motif repeats that are involved in the different loci. You can see some of the diseases. For instance, the first one, which is on top, that is amyotrophic lateral sclerosis, which involved the NIPA1 gene that is inherited in an autosomal dominant manner. It consists of 8 repeats. And when this 8 repeats usually is expanded to more than 11 repeats, then this results in the disease. And so you can have a loci that contains 8 repeats. However, some loci can go up to 34 repeats. And also the size of the expansion is also different for the different loci. So for instance, if we take this locus that is involved in oculopharyngeal myopathy with locoencephalopathy, you have in some examples the expansion of over 700 repeats when this can result in when this is resulting in the disease, whereas others there are over 1,000 repeats that can cause a disease. So how is the disease mechanisms? So this is an illustration of 3 different ways of how the disease can occur due to repeat expansion. So the first one is when you have a repeat expansion mainly in the 5' UTR region or the promoter region of the gene. When you have a normal gene that doesn't have the repeat expansion, there is normal expression of the protein. So the expression takes place and you have normal functioning protein. However, when there is a repeat expansion, usually this can cause the hypermethylation of this promoter region, which will result eventually in the repression of gene expression. So this is a phenomenon that is called the epigenetic gene silencing. The other mechanism involves the expansion of the repeats, which eventually they would recruit and sequester proteins that are involved in the splicing machinery. So when you have sequestration of these proteins that are accumulated at those repeats, that will result in having inappropriate splicing of different sets of other genes. So this sequestration of RNA binding splicing factors is then the effect of this repeat expansion. And this last example, these are the repeat expansions that happen in the coding region, which are in most of the cases the polyglutamines and the polyalanines. This eventually results in producing a protein that has an expanded motif of amino acids that is incorporated into the protein. And due to this expansion of this poly-Q or poly-A, you would have a different conformation of the protein, which eventually can result in what is this nuclear bodies that, this misfolding would result in aggregates that eventually would cause miscommunication in most of the cases in neuronal cells. So how are this type of repeat expansions detected? Conventionally, it was based on getting the DNA of an individual and then using restriction endonucleases to cut the DNA into shorter pieces, and then running this DNA on a gel that separates the DNA based on size. And after the DNA is separated, then specific probes are added that specifically hybridize to those loci. And if there is a repeat expansion, then the size of the DNA that is recognized will be larger. And this is then eventually how the data is analyzed. When you have a patient that has a specific expansion, it will be tested alongside controls that are either negative or positive. So, and based on the size, you can see that the fragments, they will run differently on a gel. And if it is expanded, the fragment will run slower on the gel. It would result in a larger fragment. And if it is negative, you would have a smaller fragment, which would run faster. And then you can compare the patient sample to the controls. And based on the results, one can conclude whether the patient is within the range of what is pathogenic or within the range of what is normal. The other method is based on a PCR method that uses uses a set of primers, one of which is has a fluorescent label, and a PCR is performed, and then the PCR fragments or the amplification product is run on an ABI machine that also separates the fragments based on size. And due to these fluorescent labels, the fragments will run on a gel, and the larger fragments would appear with a larger size. And this different peaks would then appear, and if you have a repeat expansion, you would have this particular or specific patterns that demonstrate that there is a repeat expansion, as opposed to a normal situation where you don't have repeat expansion and you have smaller fragments. So how is this done with the current methods based on whole genome or next generation sequencing? So there are many tools that are developed to specifically identify repeat expansions using NGS data. And keep in mind that the majority of NGS or next generation sequencing data that is currently produced and used in clinical diagnostics are short fragments, meaning that these fragments are usually smaller than the expected segment that is repeated, which I will show some examples of later. And there are different tools which I have mentioned here, such as Expansion Hunter, Extra, Stretch, and Thread Parse. These are all different tools that are more or less developed in 2017 and 2018. So how does it work? This is just a simplified cartoon of an illustration of this repeat expansion detector. So in the upper panel, you see an example of a segment of the DNA that contains a repeat expansion, which is depicted here in red. And then below, there is a segment of the DNA that is expected to have a normal size of the segment that is expanded in the uppercase. So usually when you do whole genome or next generation sequencing, you first fragment the DNA, which eventually is sequenced, and the sequencing fragments that is obtained, they have a specific size, which is mainly around a size of 150 or 100 to 150 nucleotides in size. And since if you execute paired end sequencing, you would have a forward fragment and its mate pair or its pair reverse fragment. So when you have a fragment that is sequenced from the forward and reverse, and this particular sequence is has been coming from the region that is not expanded, keep in mind that these fragments eventually will be aligned to a reference genome, which is below here. So if we take the first fragment, for instance, and it will be aligned to a reference genome, it will align entirely properly and correctly. However, if you take the second fragment, of which the reverse read aligns to the expanded repeat, it will, the tools that are used to align this data, this reads to the reference genome, it will attempt to align the entire fragment to the reference genome. And in this case, the forward fragment will align properly, just upstream of the repeat sequence, whereas the second fragment will align into the repeat expansion or the repeat sequence with a little bit of overhang, which is not fitting anymore. And therefore, it will be clipped. And then you have fragment 3, which is actually entirely coming from the repeat expansion. However, when you try to align this third fragment, it will align differently. And the forward as well as the reverse fragment both of them will be aligned into a specific internal segment, which is depicted in red. And the tools that do this alignment, they realize that the forward and the reverse read, they no longer follow the expected size of the library sequence that has been obtained in the first place. So as you can see, they will align differently as when they would have been aligned to an affected reference genome, which was not available. And as for fragment 4, which is more or less similar to fragment 2, it's only the reverse. So the forward sequence will be aligning to the segment that has the repeat, and then the reverse fragment will completely align to the downstream sequence. And fragment 5 will be aligned properly, accurately, also just downstream the segment. So what we have done here in our company, we have used Expansion Hunter, and we have analyzed about 45, which analyzes 54 loci that are associated with about 60 disorders. And we did this on 480 whole genome sequencing cases. And then we looked at the distribution of these repeat counts for each locus for all the cases, and then compared this to the population, the normal range in the population. So this is an example, actually two examples of two loci. One locus that is affecting FMRA gene, and the other one is affecting the AFF2 gene. As you can see, these are the data for the two, since we have two copies for the same locus in all the samples. So those two copies were analyzed, obviously, and depicted in this graph. And you see that there is some range difference within all these 480 samples. Nevertheless, it is a nice and narrow range, which is also comparable when looking at the normal distribution of the population that has data consisting of over 3,400 alleles. And the same thing more or less is true for this other locus, where we see a nice distribution, which is also very narrow, which is also more or less comparable to what we see in the normal population. For some other loci, we do see a more broader range, which is shown here in our data, which is also comparable to what has been observed in the normal population based, in this case, on 3,800 samples. And this is another example with also a broader range, which is also comparable to the normal population. So for the majority of the cases, when we looked at all these 54 loci, indeed, 41 repeat expansion regions, they were within the population distribution range. So therefore, we consider those 41 loci as analyzable. However, we also had 13 loci that demonstrated repeat expansions with a distribution that is out of range. And therefore, we considered those as not analyzable. So what can cause this non-analyzable alleles? So most of the time, these non-analyzable reads, as we are calling them, they either affect the X chromosome, and the X chromosome, as you know, in male cases, it has lower coverage. So the lower coverage may explain why they are not so well analyzable. And the other observation that we made is that those loci usually are flanked by low complexity sequence regions, which make the sequencing of those regions difficult with whole genome sequencing. And eventually, when you have low complexity sequence regions, it also results in lower coverage. So most likely, the cause for not being able to properly analyze some of those repeat regions is because of low coverage. So I would like to show this example that we have analyzed at our census of a case that is a 10-year-old female with the clinical picture that consists of cerebellar atrophy, oculomotor apraxia, ataxia, atrophy, and difficulty walking. So we did the normal routine analysis where we looked at single nucleotide variants and indels, which was negative. Then we looked into structural variants, including copy number variants, which was also negative. And next we looked into the repeat expansion using Expansion Hunter. And once we have analyzed the case, we indeed observed that there was one allele that indeed demonstrated an expansion of about 53 repeats, of which the normal range is expected to be at 22. So this was a nice example. Indeed, when we looked into the normal population, we saw that the distribution is very narrow, and the normal expected repeats is expected to be up to 31 when it should be normal. However, when it exceeds 35 repeats, then it is considered pathogenic. And in our case, there was an expansion of over 50 repeats. And if we indeed zoom in to that region that has the repeat expansion, and this is an IGV screenshot showing two samples that have been loaded. The top panel is from a control, and the lower panel is from the index case that has this repeat expansion. Now, this repeat expansion is located into this specific region. And if you look carefully, then you see that this profile at this region that has this repeat motif indeed looks a little bit different than the normal. And if we zoom in into that region, then we see indeed that there is what looks like something that is duplicated or amplified. And as you can see, the reason for this is the fact that we have this polyglutamine stretch, which has the same sequence nucleotides. And since this region is indeed expanded, and we would have in the index case more reads coming from this region that are similar, during the alignment, those particular reads that are coming from the expanded region will be aligned to this particular region, and therefore it would behave as if it is duplicated. And finally, I would just like to take a couple of, well, maybe 2 minutes to discuss the involvement of repeat expansions in cancer. A recent study was performed where in this study they have analyzed over 2,500 patients that have cancer, and they looked into 29 different cancer types. So they also run this expansion hunter approach on the samples. In this particular case, they, as a matter of fact, used the expansion hunter de novo module, which took the normal DNA as well as the tumor DNA from the same individuals. And then they analyzed all the samples together, and then they were able to identify repeat expansions in the genomes from these different tumor samples. And there were some tumor types, such as the prostate adenocarcinoma, where they were able to identify a number of repetitive or recurrent repeat expansions per sample. There were different types of repeat expansions that went over 10 times, whereas some other tumors, they were able to identify only a single repeat expansion. And the take-home message from this study is that the mutations in cancer, since the cancer has a mutator phenotype where the DNA maintenance machinery has some problems, and therefore more mutations would accumulate, and mainly these mutations also accumulate in the tandem repeats. And these repeat expansions was identified in over these 2,500 genomes, and they were also able to show that some tumor subtypes, they had specific types of recurrent expansions. And enrichments, this type of expansions were identified in regulatory elements, which suggest that they are involved in repressing the expression. And interestingly, is that one of most frequent recurrent expansion repeat, which was identified in 34% of renal carcinoma, was actually treatable by this molecule that was targeting this specific repeat. And this is interesting, as this may or may not be a possibility or an option that can also be translated to not only this type of somatic repeat expansions, but maybe it can also be used for hereditary repeat expansions that are involved in the diseases that I had mentioned earlier. So in conclusion, I would like to say that repeat expansions are genetic events that can cause disease. And in most of the cases, this type of diseases are neurological. And the phenomenon of anticipation results in that the onset is younger in subsequent generations. Repeat expansions are challenging to detect using whole genome sequencing data, or NGS in general, especially since the current or the most prevalent method of performing next generation sequencing is based on short reads. And this may change in the future once next generation sequencing will be using this long reads, because once you have long reads, these long reads will be able would have the possibility to sequence across repeat expansions, and therefore these expansions would be easier to detect with longer reads. And since there are ever improving tools to detect such alterations from NGS, it is important indeed to include this repeat expansion analysis in molecular diagnosis as a routine approach to avoid missing diseases that are caused by repeat expansions. And with this, I would like to stop and give you the opportunity to ask questions if you have any. Thank you.
Thank you very much. There, as always, lots of questions. And I start with our questions from patients and from doctors who have patients that are suffering from diseases. And thank you very much for asking. And you have a direct question how to treat your patient with a genetic disease. So please contact us directly via the website, and we will organize a genetic counseling, medical counseling session where we will directly check all the documents from your patient and then recommend treatment if it's available. So please go to us directly and we will organize an individual session for you. And thank you for your questions. So now we start with various bioinformatics questions. So the first one, why in the current literature whole genome sequencing is not recommended to detect repeat expansions?
Why? Because we do have indeed orthogonal or golden standards to identify repeat expansions, which are, I would say, easier to perform because they are done routinely for many of those regions. However, the problem is, I would say, if the physician has an extremely high suspicion that the disease is indeed caused by a repeat expansion and maybe even in a specific locus, then indeed that would be the first choice. However, since one cannot exclude that the disease can be caused by yet a different mechanism or a different mutation, therefore whole genome sequencing would be the way forward, because just assume that even that the doctor, how brilliant the doctor may be, if the suspicion is not caused by a repeat expansion, then the patient would not be diagnosed at that moment, and who knows how many other different technologies or sequencing methods or diagnostic approaches this particular patient has to go through before the final diagnosis has been reached. And therefore, whole genome sequencing does offer the most comprehensive approach for genetic diagnosis.
Thank you very much. More general question, which pipeline is used in Census for genetic data analysis?
So in genetic data analysis in general or for repeat expansions? Because for repeat
the question was in general, but you would be nice if you comment in general and also focused on repeat expansions.
So in general, we use different tools. So usually, as you may or may not be aware, that there are different sets of tools that are used at different junctions of the data analysis. The first is based on the alignment. For the alignment, we use the Dragon pipeline that does the alignment in a very fast manner. And as a matter of fact, it has been shown just recently that it's also one of the pipelines that is one of the fastest and the most accurate when it comes to the detection of single nucleotides variants. And we use also Dragon to identify structural variants as well as specifically CNVs. And for the expansions, we use the repeat expansion hunter.
Thank you very much. Also regarding this, so you use expansion hunter to identify these expansions. Does it mean that you cannot identify some novel expansions that were previously not described?
That is correct, because indeed expansion hunter focuses on specific loci that have been more or less, those loci have been used to train the tool to identify those more accurately. So we are currently restricted to using those. The problem with with identifying de novo expansions is the fact that you need also to test the tools rigorously and having also positive controls for those regions to be able to confidently call those repeat expansions in those novel regions.
Thank you very much. What is the detection rate of repeat expansion disorders in your cohort so far?
So it is, I have to admit, it's not that high, although we do have a high yield of diagnostics when it comes, if we take the majority of this, or if we take the general number of samples that we have analyzed, we have over a diagnostic yield of over 70%. As for the repeat expansions, we have, if I remember correctly, maybe a handful that we were able to identify. So it's not that many, but nevertheless, once a patient is submitted for analysis, we don't want to miss a single case. So this handful to us are nevertheless still an important number of samples that are eventually diagnosed properly.
Thank you very much. There is a general question from Doctor. So I have obtained the result from whole genome sequencing, for whole exome sequencing, and it is negative. Does it mean that my patients still have a genetic disease?
Well, I would not phrase it that way. Whether the patient has or doesn't have a genetic disease is not depending on the test that has been performed. I mean, the genetic disease may be or may not be there. An exome sequencing may not have been able to identify the disease if it was present. But it can also be that there is no disease, and regardless what test you do afterwards, which genetic test you do afterwards, you may not be able to identify the disease. So I think it's like this question is like what came first. So we have to be more or less confident that the disease is indeed genetic. If the disease is indeed genetic, based on the clinical picture, for instance, may one assume that it is very likely that the disease is genetic, and once this has been established, then one can indeed speculate that exome sequencing, if it is negative, it was not able to identify the disease, and therefore one can indeed look into other types of analyses that one can do. For instance, what I would suggest, whole genome sequencing may be the next good option to try to identify this genetic disease, which can be in a region that was not sequenced with whole exome, because maybe the disease-causing variant was not captured, since whole exome sequencing does apply a capture to be able to sequence that particular region. But it could also be that the mutation is caused by a different type of mutation that is not detectable by exome, such as structural variants, which are very difficult to detect with exome.
Thank you very much.
This made a little bit sense.
Thank you. Can repeat expansion diseases also affect organs beside the nervous system?
Can you repeat that question, please? Whether it can affect what exactly?
Can repeat expansion diseases also affect organs beside the nervous system?
I think yes. I have to admit, the clinical part is not my main expertise, but I'm certain it can affect other organs. And why? Because, as I mentioned earlier, there are some of the repeat expansion disease mechanism involve repression of the expression of some genes that are not necessarily involved in the neurological system. So therefore, the answer I would suggest would be yes.
Thank you very much. Yes, indeed was a medical question. Was is there a need to make a confirmatory test for whole genome sequencing identified repeat expansion?
Yes, that's indeed a good question. And I would say yes, because as long as, so when we perform a sequencing approach to identify genetic disease, usually the repeat expansions are a set of type of mutations that are difficult to detect in the first place with these methods. And even if you detect one, it's always it's always important, recommended to do a confirmation, just to actually establish and confirm the size of the repeat, which can be done much more accurately with the golden standards instead of using whole genome sequencing. It will give you a hint that there is indeed a repeat expansion. Nevertheless, the size of the expanded region is not that accurate, and therefore it is important to do a confirmation of this expansion, the repeat expansion.
Thank you very much. Why long-read sequencing is better for repeat expansion identification?
So, as I mentioned earlier, when I showed this this table with the different diseases, the loci, and the repeat motifs that are involved, some of them, as you could see, they spanned over, let's say, 30 repeats. So if we assume these repeats are trinucleotides, you have to multiply this by 3. So this means that you would have a region that is over 90 nucleotides. When you have a repeat expansion that, for instance, exceeds, let's say, 1,000 nucleotides, usually the Illumina sequencing, which is used by the most or the majority of the labs that do genetic testing, they have a read length that is a fraction of this 1,000 nucleotides. So if you do long-read sequencing, which is actually kilobases in length, they would span the entire repeated region. So therefore, once you have spanned the entire region, you'll be able to see exactly the size of the repeat. So with long-read sequencing, you will not only be able to identify more accurately this repeat that is expanded, but you also will be able to identify the exact size of this repeat, which is not possible when you have a shorter reads, which gives you an estimation of how much of those repeats are present in that region that is repeated.
Thank you very much. We still have more questions, and I asked the last one, but very interesting. So your lab utilizes buccal swabs at the patient sample. Is there any specific in bioinformatic analysis of the data derived from buccal swab DNA than for those that are commonly used directly from blood?
The short answer is no. Because we have, as a matter of fact, just recently published a paper on using buccal swab in clinical diagnostics, and we were able to show that there is no issues with using buccal swabs or saliva samples for sequencing with the aim to do genetic testing. The only issue that you may have is the presence of microbial contamination in the samples. However, these microbial contaminants will not affect the analysis, since they will not necessarily align to the human reference genome. As a matter of fact, you can even apply some techniques where you can use decoy sequences using frequent microbial samples, frequent microbial genomes that affect the oral cavity, and therefore you would even reduce this this misalignment better, even further. Sorry.
Thank you very much. And the rest questions we will answer online. And special thanks for your medical questions. As mentioned, please contact us directly. And thank you very much. Thank you, Najim. Have a great day. And with this, we finish our webinar.